I am Danilda Hufana-Duran, Senior Agriculturist of the Reproductive Biotechnology Laboratory, and today I'm going to share to you my experiences as a Fulbright Fellow at the University of Wisconsin in Madison, USA. I applied in the program on 2011, and I was one of those who were awarded by the Philippine American Educational Foundation with the scholarship. I got accepted at the University of Wisconsin-Madison through Dr. Jan Paris, so I went there last November 11, 2012. My field of study was in reproductive biotechnology, and the research project that I planned to conduct there was about improving the livability and fertilizing capacity of sex-sorted sperm cells by antioxidants and energy-promoting factors for the production of sex-predetermined embryos in vitro. Though I was able to carry out my research plan, some changes did occur during my study. My host professor suggested that I would have to check first the effect of the L-carnitine, which is the antioxidant and energy-promoting factor that I use, in unsorted sperm cells and in ejaculated sperm cells to establish the desired concentration before using it in a sex sperm. With that, I completed 3 studies. First, I was able to examine the effect of different concentrations of L-carnitine as supplement in IVF medium. So every Wednesday, I collected bovine ovaries and did the IBM, IBF, IBC. Cleavage and blastocyst development were examined to carry out this research. Simultaneously, I collected freeze ejaculated semen every Tuesday from ABS Global Incorporated, a private company that produces frozen semen and unsexed semen for both local and international artificial insemination works. The fresh ejaculated semen was used to carry out the research on the effect of L-carnitine on motility characteristics and membrane integrity of fresh ejaculated semen. This study aimed to examine if L-carnitine has a positive effect on ejaculated sperm cells and which concentration is the best before using it to sex sperm cells. In this study, I examined the motility characteristics of the sperm cells by computer-assisted analysis or CASA and the membrane integrity by hypoosmotic swelling test or HOS. Results in both studies showed that L-carnitine had beneficial effect but in a concentration-dependent manner, with 1 millimole concentration exerted the beneficial effect. Hence, in study 3, I used 1 millimole L-carnitine concentration in treating sex-sorted sperm cells, and the motility characteristics and membrane integrity were examined. Results of these studies will be presented in a scientific forum. Aside from the research works that I did, I also coached 2 graduating students in carrying out IVF work. I also assisted in the conduct of experiments on examining the effect of insulation on the quality of boar semen and studies on the use of vitamin E as supplement to different extenders for the cryopreservation of ram semen. I was also exposed to pregnancy diagnosis by ultrasonography. I also learned how to collect ram semen by electroejaculation. We were 4 people who worked together. 2 people restrained the ram, Dr. Paris collected the semen, while I was the one who inserted and controlled the electroejaculator. Aside from the research work that I did, I have attended some seminars and have undergone some hands-on training on the use of ultrasonography for pregnancy diagnosis in cattle and pig. Opportunities to be friended with Filipino, Americans, and fellow foreigners in the USA gave me a lot of insights. Outside the university, I was invited to deliver a talk to the PAMANA, or the Association of Filipinos Living in Madison. I delivered a talk on the topic Changing Lives Beyond Reproductive Biotechnologies, where developments and opportunities of livestock production in the Philippines were highlighted. I had the opportunity to inform them about the developments and opportunities of livestock industry in the Philippines. This aroused their interest to invest in dairy entrepreneurship as a way to help their relatives in the country. I attended a seminar with fellow Fulbrighters in Atlanta, Georgia, that provided me the opportunity to meet other Fulbrighters and Fulbright program implementers. Friendship, linkages, and international exchange plans were established. Visits to the historical museums provided a wider understanding about America, its people, and its past. Visits to the health centers provided insights on role, efforts, concerns, and programs of America on global health issues. I have initiated a collaborative research with the Laboratory of Animal Reproduction, University of Wisconsin-Madison, on the use of Fourier harmonic analysis in predicting water buffalo fertility. I have prepared and submitted the project proposal to PCCR in the unit for review and necessary actions for funding support. This Fulbright program also provided me the opportunity to materialize a long-planned collaboration with the Instituto Nacional Autónomo de Investigaciones Agropecuarias or INIAP in Guayas, Ecuador. I was invited by INIAP to train their staff on reproductive biotechnology, and we have developed partnership and technical exchange on the implementation of reproductive biotechnologies for livestock improvement. The Fulbright program provided me the opportunity to see and visit several states in the USA, been at the Sears Tower in Chicago during the New Year, and stayed with my niece in Indiana during the New Year holidays that enabled me see Michael Jackson's childhood house. Been in the Houston, Texas to serve as honorary guest during the 40th founding anniversary of the Kappa Epsilon Alumni Association of North America. And with this, I had the opportunity to visit the NASA and the Liquid Church of Joel Austin. And my friends drove me to the Louisiana to visit the place. Attended Fulbright seminar in Atlanta, Georgia, and visited the Los Angeles and Long Beach, California for a farewell party and pleasure trip arranged by the KIANA or Kappa Epsilon Alumni Association of North America. Visited the Hollywood, the Universal Studio, Griffin Observatory, and ate at Acapulco and Queen Mary. These trips allowed me to see at least the aerial view of Detroit in Michigan, Minnesota, New York, Minneapolis, St. Paul, and Alaska. The trip to Ecuador provided me the opportunity to see Panama. Indeed, I could say that my Fulbright program in the USA was a fruitful and very fulfilling and wonderful one. And I would like to thank the Philippine American Educational Foundation, the Philippine Carabao Center, and the University of Wisconsin-Madison for the opportunity that they have given to me that helped me further my professional growth and experience.